hopefully today I will demonstrate how live cell imaging has transformed our day-to-day research on foot and mouth disease virus. So I just want to give a little overview here on what the webinar will contain. So I'll give a brief introduction to foot and mouth disease virus and the purpose of our research to try to put everything into context. I'll then go on to talk about how and why we use the IncuSite to measure FMDV replication in mammalian cells. Following on from that, I will discuss resources that have been made available to us from Essen, such as software upgrades and reagents which help with our day-to-day analysis. And then I'll finalise with a brief overview of how we set up experiments and export the data. So FMDV is a member of the picornavirus family. It is a non-enveloped icosahedral positive sense RNA virus, and common examples of picornaviruses include polio and the common cold, which I'm sure a lot of you have heard of. And for foot and mouth disease, 7 distinct serotypes exist. A and O, which are the most common, C, which is almost non-existent, and Asia 1 and South African territories 1, 2 and 3, and thousands of strains exist within each of these serotypes. The genome consists of 2 untranslated regions which contain structural elements which are important for replication and translation, and the viral polyprotein is translated as a long open reading frame and consists of both the capsid proteins and the proteins that are responsible for replication of the genome. And these are processed into the 14 individual proteins by 3 viral proteases, the 3C, the 2A and the L protease. So the disease that is caused by FMDV, it causes lesions or blisters inside the mouth and on the teats and on the hooves of clothed and hoofed animals such as pigs, sheep, cattle, etc. Outbreaks are very detrimental to the economy. So using the 2001 outbreak as an example, it cost the economy approximately £8 billion, which was mainly due to a decrease in tourism rather than in agricultural costs. And during this outbreak, 6 million animals were slaughtered, most of which were healthy. So the current disease control strategy within the UK and Europe, it involves culling of infected or susceptible animals, inhibition of animal movement and ring vaccination in extreme circumstances. And this disease is endemic in many developing countries, meaning that it's still a threat to disease-free countries like the UK through trade and tourism. So just wanted to give you guys an idea of where outbreaks have actually occurred within the last 6 months. So the countries listed here in green are countries which are disease-free through vaccination or without vaccination. And those in grey are countries which they are endemic or don't have an official status for foot and mouth disease. So within the last 6 months, there have been outbreaks occurring in mainly the eastern side of the country, of the world. And just last week, there's actually an outbreak in Cambodia, which is still ongoing. So the current vaccine for foot and mouth, it's a killed preparation, which requires bulk growth of virulent virus within suspension cells in large vessels, which is then chemically inactivated. And this is a problem when it comes to disease security, because there is the risk of virus leaking from production facilities and causing outbreaks. The vaccine also requires repeated administration because the immune response that is elicited is very poor. And also there is no cross-protection between the 7 serotypes, and there's also sometimes not cross-protection between the strains within each serotype. It also requires crew purification. So the vaccine prep can be contaminated with non-structural proteins, and this is an issue when it comes to something called DIVA, which is differentiating infected from vaccinated animals, where in a vaccinated animal you should only get antibodies against the coat protein, whereas in an infected animal you should get antibodies produced against all the viral proteins. But if the vaccine prep has contamination, then it's difficult to do this, which is an issue when you're trying to control through an outbreak. So because of the current caveats with the current vaccine, what we'd like to do is we'd like to make a new live attenuated vaccine. And the reason for this is because there's a certain number of pros that would help solve the issues with the current vaccine that exists right now. So these are things such as biosecurity. So bulk growth of attenuated virus isn't a risk if the virus escapes from a production plant. It's much easier to administer than a killed vaccine, which requires an injection. And also the immune response is far superior in comparison to a killed vaccine, which means that you should not need boosters required. So the objectives for us, is one of which is to produce a new live attenuated vaccine. However, to study foot and mouth, it is a SAPO level 4 pathogen and requires high containment facilities to do any research. So to get around this, what we have done is we have made a fluorescent replicon system which is non-infectious and biosecure. And what the main objective I'm going to cover today is, can we study FMDV RNA replication via live cell imaging? Because discovering more about how the virus replicates will help us in the development of a new live attenuated vaccine, as not much is known about the way that FMDV replicates in comparison to the other common coronaviruses. Okay, thank you, Amanda. So it's good to know that quite a lot of you are virologists, because that does help in the next part of my talk. So I just wanted to go over the classic virus rescue the classic way that we rescue RNA viruses through reverse genetics. So it involves reverse transcription of the viral RNA into complementary cDNA. And this complementary cDNA is cloned into a plasmid vector under the control of a mammalian promoter. And what you can do is you can either transcribe this cDNA into transcript RNA, transfect this into a cell, or you can transfect the cDNA itself into the cell, and then you can rescue viruses. And what you what you can do is you can modify the cDNA and then rescue the virus and look at the phenotype from this modified cDNA. But obviously we cannot work on an infectious agent, so how we get around this is by using the replicon. So we obtained this replicon here, which we call PT7rep. It's from the O serotype, which is the most common serotype that's circulating around the world just now. And this replicon, it had the chloramphenicyltransferase reporter protein in place of most of the capsid proteins and of the L protease. So replication could be monitored via cat assays of cellular extracts. However, this is quite an insensitive and laborious process. So what we did was we reinstated most of the L protease, as we want to have a viral genome that is as much like Wildtype as physically possible. Then we replaced the cat protein and most of the remaining capsids with a GFP. So this GFP is actually called Telasarcus GFP, which I'm sure quite a few of you haven't heard of before. And the reason why we use this GFP is because, one bonus is it's actually brighter than eGFP, which has helped with our detection. But also we discovered through our initial IncuSite experiments that it seemed like the eGFP was actually being cleaved. And through further experiments we realized that the L protease actually cleaves eGFP, so we had to find a suitable replacement. So we used this Telasarcus GFP, which is from the C feather, and it is not cleaved by L, so it's good to go. The next replicon we wanted to make was a negative control. So this negative control has 2 point mutations within the polymerase. So the active site is mutated from GDD to GNN, rendering it unable to replicate its own genome. And what this does is, so we have a wild type of negative control, and the negative control allows us to measure how much fluorescence is generated through input RNA versus a wild type replicon that can actively replicate. So when we take this into the context of what I was discussing before, essentially most of the steps are similar to classical virus rescue. It's just we use a replicon, there's no capsid protein, therefore no viral particles. So rather than do virus rescue and do plaque assays, etc., what we do is we can modify the replicon and look at the phenotype through live cell imaging. So RNA transcription, for those of you who are non-virologists, what we do is we take the replicon, we linearize it with a unique restriction enzyme. We then, so this has been a little bit slow, we can then reverse transcribe this into transcript RNA using the T7 RNA polymerase, which fires off the T7 promoter, and then we can take this transcript RNA and we can put it into mammalian cells. So we make liposome complexes from transfection mixtures. We then transfect these into cells. The viral RNA is released into the cytoplasm. It's then translated by the host cellular ribosomes into all the viral proteins important for replication and translation, but also GFP, which is then fluorescent when excited by the appropriate wavelength of light. And this is measured by the incucyte, which lives inside the incubator. So this is automated, and it's done continuously, so you can just set up your experiment, pop it in, and walk away from it. Just to give you an idea of what the data looks like, so this is the wild type and this is the negative control here. I've got some videos. These are baby hamster kidney cells. This is using the 10 times objective. There are 3 objectives available, the 2, the 4 and the 20, which we have all of, but we do most of ours using the 10 time objective. And hopefully what you'll see is there's differences in fluorescence intensities between the wild type and the negative control. And you'll also see quite a lot of cytopathic effect as the L protease shuts off host cell translation, which causes the cells to die. So this is the time post-transfection here down in the right-hand corner. And hopefully, like I said, what you can see is there's a clear difference between our wild type and our negative control. So when you take this data, which can also be exported as images as well as movies, you can analyze the data and plot it, and what you see here is this is the wild type, which is replication competent, and then we have our negative control, which cannot replicate and is a measure of what fluorescence is derived from the transcript RNA that we transfect into the cells. So any modifications that we make to the genome which would affect replication when we go to study this would hopefully lie somewhere in between these two forms. And because the incucyte zoom can also measure red fluorescence, what we decided to do was we made cherry replicons. So again, I'll just play the video so you can just have a look and see. So this is BHK cells again. And as you can see, the cells go nice to the red, they round up, they die. And one of the main reasons we wanted to make these replicons is because we want to undertake research that involves transcomplementation, where you would modify the green replicon in a certain way that you think it would affect replication, and then you co-transfect it with a wild-type red replicon as a helper. And that's actually research that's been done quite a lot by our collaborators at Leeds. So that's the system we use, that's the forms we use. We have green and red replicons, we have replication competent and incompetent replicons. Now I want to go into resources that have been made available to us by ESSEN, which actually helped with our day-to-day research. So one of those, which fits in nicely with the cherry replicons I've discussed, is something called Coincident object analysis. So our collaborators at Leeds are doing a lot of transcomplementation assays with green and red replicons, and they contacted ESSEN to ask if it was possible, if they could design a new software feature where we could tell whether cells were both red and green. So again, like I said, working together, our collaborators at Leeds provided ESSEN with images and data, and they actually designed this new software feature for us, which is brilliant. So I've just given you a little example here of what some data can look like. So this is an experiment I did where it's a wild-type green replicon with a replication deficient red replicon. And as you can see here, so with this coincident object analysis, you can get the number of red and green cells and yellow cells, and also you can look at the area that is covered in the well by red, green or yellow cells. So as you can see here, this is how many cells that are green, this is how many cells that are red, and in yellow here, this is the cells that are both yellow and red. Sorry, yellow and green and red, sorry. So here, this is just a representative image of 4 hours post-transfection, which sits about here, and hopefully you can see that there are cells which are just green and are just red and are a combination of both. So this has greatly helped our collaborators in Leeds, and we are also using this for another project that I'm not going to talk about today, which is involving the development of a DNA vaccine, and this will help us greatly with our research. So another reagent that is available is a lentivirus construct. There's 2 different types. There's red and there's green. What you can do with this is you can make stably expressing cell lines which have either RFP, well, a form of RFP called MK2, or GFP, which are expressed in either the nucleus or the cytoplasm. So we have both green and red for the cytoplasm and the nucleus, and we also have them in HeLa cells as well as BHK cells. So I've shown just a representative image here of a nice fibroblastic BHK cell with a red nucleus. And what this does is this enables us to monitor cell counts during the course of an experiment, and also allows us to calculate transfection efficiencies between experiments, so that between individual experiments we know that we're getting the same amount of cell counts but also the same transfection efficiency. And again, just got a little video here to show you what this looks like. So as you can see, the cells that are transfected, they turn green, they round up, they die, but you can also see there are a number of cells here that are not transfected. So again, when we can plot this data, so this is just the red count that I'm showing you here. So this is the mock, which, as we would expect, keeps going up and up and up as the cells grow, because it's just mock transfected. But those cells which are transfected with the replicon, which round up and die, the cell counts start to die off. And again, as you can see, we've managed to seed the similar amount of cells within each condition. So these, then I can take this data and I can take the green counts and I can get an idea of transfection efficiencies, which I haven't showed you. So these resources have really helped us day to day when it comes to our analysis. So hopefully I've managed to show you what the system is, what we use, how we use it, the reagents that are available that also help us to do our research. So what I wanted to then just briefly mention is the experimental setup and the data export using IncuSite. I'm not going to go into the data analysis, just in the interest of time and the scope of the webinar, but I just want to essentially let you guys see how easy and quick it is to set up an experiment and export your data. So this is what the user interface looks like. And if I'm just going to focus on the main elements of this. So the drawer setup here, which essentially looks just like how it's set up inside the IncuSite. So there's 6 places available, and you can use dishes, flasks, or tissue culture plates. And here I've just shown you a 12-well plate, as that's what we use routinely. So it's as easy as clicking on the appropriate position, adding a vessel, and then a pop-up box will come up, and you can choose your vessel from a wide range of distributors and different well contents. And then what you can do here is on the side, you can select your channel, so phase, green, red, combinations of either. And over here there's a drop-down box which allows you to scan your wells differently depending on how many images you want to take. So again, routinely we choose 9, but you can change this depending on the tissue culture vessel that you have added. And the next thing I want to show you is just this Properties tab here. So the Properties tab is where you can add an experiment label so that when it is archived, you can go and you can find your experiment really easily. You can also label the cell type, the passage number if you want to, and also add something called a plate map, which I won't show you, but essentially all it is a label so that you can know what exactly is in each well in the conditions that you're testing. The next thing is to set up an interval. So it's as easy as you right-click on the timeline up here, and then you just fill out these boxes. So you put in the time you want to start your experiment at. So I've said 4:30. The intervals that you want to do. So how often do you want to add these scans? So routinely, again, we do every hour. And we tend to run our experiments overnight. So we just run them for 24 hours, but this can vary. You can run it for as little, as often as you like. And then all you would do is click apply, and that's it. It's ready to go. You can walk away with it and get on with other experiments, etc. So when it comes to, not analyze your data, post-analysis of your data and exporting your data, again, it's as simple as, so this again is the interface. You can select the start and end time of the data that you want to export. You can also select which wells you want to export and put onto your graph. If you want to include replicates, which is something to do with the plate map. So the plate map can tell you if you have replicates and it will pull them through into your data export. Or you could do columns, or you could do rows. And it's, again, there's a microplate graph here as well, which shows you little graphs of every single condition that you have in your well. And if you want to export your data, it's just as easy as clicking on this box down here, and you can export your data into GraphPad or Excel. But you can also generate graphs using the analysis software, which I'll just quickly show you. So this is what the graphs look like that you can generate. And these are really great for things like lab meetings, and also if you want to keep a record in your lab book, so you can just print these out and stick them in. So I just want to finish with letting you guys know what life was like before we had the IncuSite, and essentially how unfeasible it was for us to carry out our research without it. So what we did initially was we used a combination of a benchtop fluorescent microscope and a plate reader. So what we would have to do is, at every time point, we would have to take the plate from the incubator and put it onto the microscope or into the plate reader. We would then have to acquire images manually because our plate reader didn't have a camera. And then we would have to take these images and analyse them using imaging software or export readings from the plate reader. Now, this generated a lot of problems, so we're just going to highlight them all here. So because the plate reader had no humidified or CO2 controlled environment, which meant that we had to keep removing the plates. Also, there was no pH control, which was an issue with media. It's also very invasive, so you are constantly disrupting your experiment and therefore your cells. It also generates a lot of temperature changes, which can affect your experiment. When you acquire the images, you cannot define the part of the well that you want to take the image from. So there's a lot of experimental bias, because each time you take a time point, you will not be taking an image from exactly the same point in the well each time. It's also laborious and very time consuming, which then hindered the amount of time points that we could take because we had to do it manually. Whereas with the IncuSite, you just pop it in and you can walk away, which frees up so much time. And also, when it came to the analysis, it was very limited. So with the IncuSite, you can plot different forms of fluorescence. So you can do the average fluorescence, the number of cells that are green, the total fluorescence, as well as many others. Whereas with the plate reader, all we could do is have essentially a total fluorescence. So that limited our analysis somewhat also. And there was a low signal to noise ratio, so it was very difficult to actually detect our cells above background because the media autofluoresced so much and we just couldn't get the background down at all. So that's an idea of what it was like before, and then I hope you can agree here that with the IncuSite, it's just made things so much easier for our day-to-day research. So in summary, hopefully I've been able to show you that the Replicon system that we use in combination with live cell imaging provides a rapid facile screen for RNA replication. We can look at the differences between a wild type and negative control and distinguish those differences. And also we are further in our research by generated attenuated replicons, which we can then measure against these wild type and negative controls. And again, how the IncuSite has transformed how we carry out our analysis day to day. And just to let you guys know that obviously I've talked about it in terms of replication, but one other aspect that we're doing as well as looking at attenuation and replication is antiviral screens, as well as, so this work is part of a huge collaboration between 5 different universities. And I've just highlighted in green here what research is actually going on at these institutes, and every single form of investigation essentially in the end is to try to find a new live attenuated vaccine or ways that we can change the way the disease is controlled. And University of Leeds, which I've mentioned already, and ourselves, we are the 2 sites that use the IncuSite, and essentially our work has helped the other sites with what they're carrying out as well. So this isn't just me, this is part of a much bigger picture. So I just want to finish by acknowledging everybody in the consortium, Essen for supporting the webinar, Bite Size Bio for allowing me to do the webinar, our funding bodies, the Wellcome Trust and the BBSRC, which are both UK-based funding bodies, all the colleagues within my department, a lot of past members from our lab who have been involved in a lot of this work and others who have not. And then I just want to finish by letting you guys see what the present members look like. So this was actually a thank you card that was generated by one of our recent project students. So Gary and Sean are both postdocs as well as myself, John our technician, and then Martin, who is aka the boss. And if any of you could or no Martin would agree that this is actually a very good true likeness of this. And also, I've just put up here two publications of ours that would help with further reading if anybody's interested, as well as the ResearchGate profile for myself, which lists some other publications which you might find interesting, as well as the website here, which is part of the consortium and lists exactly what we're doing as a consortium.
Thank you very much, Amanda, and thank you, Fiona, for a very stimulating and interesting report or summary of your experience with IncuSite. Much appreciated. So what I'd like to do for the next 10 minutes or so is really expand a little on the topic of the webinar, so furthering insight and productivity in cell biology with real-time quantitative live cell analysis. Just to begin with, I think it's useful to perhaps just redefine or re-explain for those of you who may not be so close to this field what we mean by this term. So continuous live cell analysis is a continuous or at least semi-continuous cellular measurement where we monitor, make relevant analyses of parameters over time and in real time of cellular function, cell morphology or phenotypic behavior. And some of the key attributes are that these measurements are made from living non-perturbed cells. So in this case, environmental control is key. All of our measurements are made from within the stable environment of the incubator. We try not to wash cells. We certainly don't fix and stain them or do anything else that potentially could introduce artifacts in measurement. So everything is done from living non-perturbed cells. The measurements that are made are non-exhaustive, so they do not deplete the cells throughout the course of the measurement, so you're free to use the cells for other experiments at the end of the study as you wish. And if the biology requires you to make long-term measurements from cells for hours, days, and potentially even weeks, this methodology is entirely amenable to that because, as I explained, the cells stay within the controlled environment of the incubator throughout. An important point that I'd like to make, and Fiona illustrated this in a couple of her examples, is that continuous live cell analysis is applicable across workflows, whether you're culturing or performing husbandry on your cells when you're manipulating them, transfecting them, and through to the more conventional arm of the assay that you might think about for your usual cell measurement. And I'd like to just expand on that point in the next slide with the following visual. So in this, I compare and contrast the workflows which I hope you will all recognize, irrespective of what field you're working in. If you're working in cellular measurement, you almost certainly at some point will be harvesting cells, you will be culturing them, you will then perform some kind of manipulation. It may be a CRISPR manipulation or a gene transfection or a treatment with small molecules or antibodies, and then ultimately you'll start to measure the impact of those manipulations on your cells of interest. Now, if you're using traditional endpoint measures, typically you may make a qualitative assessment through the culture and manipulation phases. You may look at the cells under a microscope to verify that they look well, but you're unlikely to be making any quantitative analysis. In contrast with the continuous measurement, you will be taking images, quantifying cell morphology, cell growth, cell behavior throughout the entire continuum of this workflow. And when you get to running your kinetic assay, you will be taking repeated images as opposed to the single arbitrarily defined endpoint. You'll be taking repeated images and repeat analysis until you're comfortable you've learned everything you need to learn from your cells and you're comfortable that your experiment has truly come to an end. So I think that's clear to see what value the continuous analysis approach provides compared to an endpoint measurement. Just to perhaps expand on and give you some specifics around this culture manipulation and assay workflow. So if you are, when you're thawing cells or harvesting cells or subculturing cells, it's very valuable to be able to monitor them and check that they're healthy and happy. When you're doing things like stem cell reprogramming or differentiation or clonal dilution, these are long-term processes where continuous monitoring can really inform you of when things have happened and what that looks like. One of the kind of most routine and common uses for continuous live cell analysis is simple cell proliferation assays, which really cut across when you're culturing and then manipulating all the way through to the assay itself. With live cell assays, people are using phenotypic assays more and more routinely nowadays, both for screening and for mechanism of action studies. So the IncuSite way, our way of continuous live cell analysis, Fiona explained very nicely, just to again recap on some of the key points. So the IncuSite lives constantly in the incubator, and it is coupled via an externalized drive to your network, so you can remotely view what's happening to your cells from your office, or potentially even from home if you can communicate to your computer network. It's an automated time-lapse imaging approach. Fiona showed both label-free and red and green fluorescence analysis. Importantly, almost all of the measurements you'll make will be using your own regular tissue culture consumables, whether they're microtiter plates or flasks or Petri dishes. We do offer a number of specialized microplates for different types of applications, but the vast majority of work is done with completely regular tissue culture consumables. I'll come on and talk a little bit more about applications on the next slide, but again, an important point I'd like to share that Essen as a company, we're certainly not interested in just providing into the market the hardware. We're very much interested in providing integrated solutions for researchers, which is to couple appropriate software to the hardware, and Fiona nicely introduced a couple of reagents and consumables, all of which are geared and tuned and formulated towards this type of kinetic, non-invasive live cell imaging approach. So very much our thought processes are to provide integrated solutions for researchers working on different cell-based applications. Okay, so this, if you like, wall of applications illustrates a number of different things that can be done using our platform technology. Just to call out a few, and to note firstly that a number of these measurements are made entirely through phase contrast imaging, so without any fluorophores or fluorescent proteins whatsoever, things like proliferation, cell invasion, chemotaxis, clonal dilution. Several we use non-perturbing fluorescent protein constructs to track cells in co-cultures, so things like the angiogenesis assay and the neurite outgrowth co-culture with neurons and astrocytes. And then a number we use non-perturbing small molecule probes, such as the caspase substrate and an annexin binding label for kinetic apoptosis assays, as well as cell impermeant DNA binding fluoroprobes for measuring cytotoxicity. So Fiona's viral RNA replication example is not specifically called out here, but I guess it would come under the family of gene transfection measurements shown there there in the middle. Okay, and then just to orientate these in a slightly different way, this will be the incarnation of our latest website, where we've clustered these applications neatly under these three broad categories of cell health and viability, cell monitoring and workflows, and then the phenotypic assays which I introduced. Probably the immune cell killing is one of the most popular at the moment, as you would imagine from the upsurge in interest in the immuno-oncology field. Okay, so just to spend one slide each on the two main points of the title of the webinar, so furthering biological insight and enhancing productivity. Fiona showed you a few movies. I'll just show you one or two others to really try to reinforce this point of seeing is believing and is understanding. So on the left here, I'll show you first a movie of some J774 macrophages phagocytosing some Frodo green labeled bioparticles. I'll speak as we do this. So what we are seeing here are these specific cells ingesting the bioparticles, and when they do so, the labeled bioparticles go into the acidic environment of the lysosome, and then they start to glow green. So simply by watching the move, we get a very clear insight into the coupling between the appearance of the fluorescent signal and the phagocytosis of the cells. We can see them physically digesting the bacterial bioparticles, which makes it a very credible measurement. We also learn other things about how the cells divide, how they reduce motility as they start to almost overfeed and eat too many bioparticles, and some of them then start to die, having almost eaten too much. So all of those things are insight which would not be gathered by either static endpoint imaging or by non-image based assay technologies. The second one is an example of chemotaxis of HT1080 osteosarcoma cells towards serum. So this is an experiment done on a specialized Clearview microplate, and what we see here are AC1080s cells disappearing down these etched microholes in the plate towards serum on the underside of the plate. So we quantify that by analysis of these phase contrast images. So again, it's very credible and very clear to see that the cells physically are moving down the holes. We can make descriptors of their morphology, we can see the cells dividing, and a number of other insights that would not be gained by endpoint or non-image based analysis. Now, of course, time-lapse imaging is done and available to all by a number of other microscope methods, and the types of movies I just showed you could do by other systems. A main point of differentiation for our approach is the scale at which this can be done with IncuSight. So we enable up to 2,300 distinct parallel time-lapse movies. If you put 6 3/8/4 well plates in at a time, that's the type of thing that you can do, which is obviously orders of magnitude more than can be done on an inverted microscope system. All of the image acquisition and analysis is done automatically. So as Fiona again explained nicely, you can set up an experiment, you can program the IncuSight to do its thing, you go away for the weekend, and when you come in on a Monday morning, all of the data is analysed and displayed in the types of visuals illustrated here, these microplate views where you can immediately see the time course of the biology of interest and the differences between your control treatments and in this case your drug treatments for this immune cell killing assay. Another important point is done with on-the-fly decision making. So if, as your experiment unfolds, you see something that you feel leads you towards making a different decision or doing something that you hadn't otherwise expected to do, you do that in the full knowledge and information that the data is analysed and is there in front of you. So you don't have to wait for everything before you can really understand what happened. Of course, as with all experiments, not all experiments work, but again, the elegance of the continuous live cell approach is that even when things don't work out as you'd hoped, you can generally see what went wrong and when it went wrong, and this really helps you to reduce the amount of rework that's required and to minimize assay failures, and all of this contributes to enhancing productivity. Okay, so just to finish up with a slightly different angle. So there are now in excess of 800 scientific publications describing IncuSight data. I take this as an opportunity to direct you towards our publications portal on our website. You'll find Fiona's publications on viral replication there, amongst many others, so it's a nice searchable format. You can also see where the geographical distribution, if you like, the demographics of these publications. So you may find labs nearby to your own that may be publishing on IncuSight where you may know a friendly face that you can talk to. So it's a nice vehicle to find out more about who's doing what with the platform, as well as hopefully to build confidence in this particular field.